Hello everyone, I'm Aksha from Final Year Biotech. I'm going to take a topic of yeast to hybrid system. What is yeast to hybrid system? It is also known as yeast to hybrid or two hybrid screening. It is a molecular biological technique that is used to discover protein and protein interactions and protein DNA interactions. It tests for physical interactions and it binds between two proteins or a single protein and a DNA molecule respectively. So now history. This yeast to hybrid system was first pioneered by Stanley Fields and O.K. Curzon in 1989. It is used to detect protein-protein interactions using Gal4 transcriptional activator of a yeast Saccharomyces cerevisiae. This Gal4 protein activated transcription of a gene involved in galactose utilization, which formed the basis of selection. And it describes many alternative methods which can use different host organisms instead of yeast, such as E. coli or mammalian cells. Principles. Yeast to hybrid assay relies on the expression of a reporter gene such as LAC-Z or GFP, which is activated by the binding of a one particular transcription factor. This particular transcription factor is composed of DNA binding domain and activation domain. This binding domain, which is fused with query protein of interest, is known as bind, and the activation domain, which is fused with protein library, which is known as prey. When the reporter gene expression is activated, a transcriptional unit must be present at the gene locus, only possible if bait and prey interact. Now the protocol. The early yeast to hybrid systems were based on the finding that many eukaryotic transcription factors have separable DNA binding transcriptional activator domains. As you all know, this activation domain is such in the case of his gene, and the DNA binding domain is in the case of UAS. And the bite, which is the protein of interest, is fused to a DNA binding domain. The proteins that bind to bite, it is fish or prey, or fused to a transcription activation domain. This DNA binding domain is bind with bite, and the activation domain is bind with fish A or prey. Proteins that do not bind to the bite will not activate the transcription of a reporter gene, that is his in this case. Any protein that binds to the bite will activate the transcription of the reporter gene. And this transcription initiation complex is binds to the mRNA or his protein. Now the applications. It detects drug and poison discovery. It identifies novel protein-protein interactions. It can also identify protein cascades. Can only identify interfering proteins in known interactions. Reverse to hybrid system. It determines sequences crucial for interactions. And advantages. Immediate availability of a cloned gene of interacting protein. Only single plasmid construction is required. Interactions are detected in vivo. Weak transient interactions are also can be detected. Protein purification is not necessary and no antibodies also not required. It can accumulate a weak single over time. And this disadvantages. False positives are the largest problem with the yeast to hybrid system. Possible incorrect protein folding in yeast. Gene encoding target protein must be available. Fail to detect some no interactions. Fusion proteins may inhibit the virginal interaction. Now, let's see about the examples of uses of the yeast to hybrid system. It identifies cas-based substance, interaction of calmodulin and L-isoaspartyl methyltransferase, genetic characterization of mutations in E2F1 peptide hormone receptor interactions. This is the overall concept of yeast to hybrid system. Thank you.